if you click in the upper right corner, there is a do not disturb. The very upper right, to the right from your search button.
All right, Greg, we're live on YouTube, so you can go ahead and get us started.
Okay, well, welcome everyone. So it's a pleasure to have our panel and our two speakers today for this KNAW biophysics meeting. Our two speakers are Leonid Murney and Agnese Seminara.
Right, Greg, live on YouTube, so you can go ahead and get us started.
Oh, sorry, I'm getting some feedback. Let me fix that. We will start off with Leonid. Both Leonid and Agnese will give 15-minute talks followed by 15 minutes of discussion for each. At the end of these talks, there will be a tutorial session with Leonid. So without further ado, let me introduce our first speaker, who is Leonid Murney. Leonid received his PhD in biophysics from Harvard University. After a few years as a junior fellow at Harvard Society of Fellows, he was appointed to the MIT faculty in 2001, joining the MIT Health Sciences and Technology Division and the Department of Physics. His research combines first principle physics with the analysis of genomic data. His efforts in biological physics involve the development of novel computational tools and physical models of genome organization and dynamics. He's also a fellow of the American Physical Society, an associate faculty of the Broad Institute, and an associate member of the Dana-Farber Cancer Institute. It's a pleasure to have Leonid with us today. He will talk to us on the biophysics of genome editing. Leonid, take us away.
Okay, genome folding. Thank you so much for the introduction. So I will talk about biophysics of genome folding. And so originally I thought about giving a short talk about the biophysics of genome folding in general, and then going into tutorial connecting this to function, but I decided to swap the two. So I will give a very quick introduction now and focus on connecting genome folding to function. So sprinkling in enough biology now, and then over tutorial we'll go more slowly over different aspects of biophysics of genome folding. What's the problem? The problem is that DNA, as you see here on my slide in this toy that we were playing with in the lab. So DNA are exceedingly long molecules. So the length of DNA in every cell of your body is 2 meters, and multiply this by the number of cells and by the number of participants on this call. This gives a few light years of DNA, which is striking, I think. So that like 20 human beings contain a light year of DNA. But in every cell, there's a couple of meters that need to be compacted in relatively small volume. And the question is, how is this working? What's the mechanism of this compaction? And how this compaction actually allows genome to work? That's a really challenging question. So the main source of information about how the genome is folded comes from the data that are called high-C data. So high-C data are on the left here. These are maps of chromosomal interactions. So every pixel here, so it's a map, genome by genome in length. And the intensity of every pixel tells you how frequently these two genomic regions are touching each other. And so these maps are incredible in their resolution and dynamic range. So for example, the squares that you see here on the left are chromosomes. So I will just play this animation where on the left I'm zooming into this map of high-C map, and on the right I'm proportionally zooming in Google Maps, and you'll see that one high-C map of the genome is actually a Google map of the Earth at about 50-meter resolution. So every high-C map is a Google map is a Google Earth. So it's an incredible amount of information from which we're trying to get an idea about the physical mechanisms underlying genome folding. And I will talk more about this in my long tutorial. Now I will focus on one element of this organization. So if you zoom into this, into the diagonal of this map, you'll see structures close to diagonal, and they're called topologically associating domains. And what's important about them is that these are like squares. I'm not sure you see. Can you see my mouse? Anybody can tell me? Is my mouse? Yeah, okay. So these are the structures next to diagonal, the squares next to diagonal. So these are regions of enriched contact probability. So as if the genome is like locally crumpled into local crumples, and they're kind of isolated from each other at the boundaries of such crumples. And the question is, what kind of mechanism folds genome into this organization, this kind of block diagonal structure? And the second question is, how is it related to function? And I will now kind of basically make a provocation suggesting that this mechanism is very important for function, and so try to get some feedback from it. So we worked on the problem trying to understand what mechanism leads to this folding, and proposed a mechanism and ruled out many possible mechanisms and focused on one that did reproduce these features, and I will show you examples in the next slide. And the mechanism is the following. So the idea is that, and this is, we're kind of in 2015 now on this slide, so we were trying to collaborate with John Marco on some other projects, and John has suggested this idea, a fellow physicist from Northwestern University. John suggested this idea that there are machines that land on DNA and extrude progressively large loops. And so, okay, that's interesting. So that would be great for compaction. And that's, in fact, this idea has quite some history that this might be a mechanism of chromosome compaction. But what we were saying, what if the same mechanism works during interphase, and this loop extruders land on DNA, form progressively larger loops. When they fall off the DNA, the loop dismantles. When they meet each other, they block each other. Okay, these are the rules of the game that you need to set up for this model to work. In addition to all of these rules, we say, in addition to all of this, there are also boundaries. And if there is a boundary, when this machine comes to the boundary, it would stop one of its motors and would continue reeling DNA in on the other side. So that's the idea. And the question is, can this reproduce this TADs, the structures that we've seen in high-C maps? And what simulations have shown us, so we set up large computer simulations of a polymer system subject to to activity of the loop extruders with boundaries. And what we found in simulations is that this indeed gives maps that look maps of contact frequencies. So these maps look remarkably like maps that are obtained experimentally, like real high-C maps. So these maps have this block diagonal structure, they have stripes, they have dots. So this is, I will just show some examples of this high-C maps. Again, they have the stripes, dots, and squares, and nested squares. So these maps look remarkably like maps seen in high-C. So with that, we said, okay, so that's nice. Maybe that's a real mechanism. How do we know this? So we also proposed that the loop extruders are cohesins, molecules of cohesin, and again, known to be associated with chromosomes, but not known to be motors. In fact, the idea that cohesins and generally proteins of this class are motors was met with quite some resistance in the field. But we're saying, so they got to be motors that extrude loops, and then CTCF molecules would be boundaries. And this theory was tested by several papers. So these are just 6 papers that I think are the most influential, where experimentally people perturbed either part of the extrusion machinery or CTCF's boundaries. And again, in all of these experiments, the results were basically consistent with the theory, saying that if you remove, so on the left I will show removal of loop extruder, the motor. In the middle I will show removal of the boundary, and again, both cases we expect to see domains to disappear. And on the right we'll see what happens if you put too much motors on DNA. And again, as expected, in this left 2 panels, in the left and the central panel, all the structures disappear. On the right, you see enrichment of all of these features. And I will not go into details, but sort of we wrote reviews about that. So generally, we see that experiments strongly support loop extrusion by cohesin and stopping by CTCF. Again, that wasn't obviously the whole story because nobody have seen the actual process of extrusion. And again, several groups, starting with the brave experiments in the lab of Kees Dekker at the Technical University of Delft, where Kees's group have put a piece of DNA, stretched it in the flow, and then observed condensation to do loop extrusion. And then since then, several groups have did similar experiments with other molecules in this class. And particularly surprising was cohesin, and that's the experiment that I'm showing on the right. So again, it's a similar setup to Case-Decker's experiment. So this is done in the group of Jan Michael Peters in Vienna. DNA stretched in the flow. So here you see DNA, it's stretched in the flow, it forms an arch. And then the molecule of cohesin binds, and then gradually it starts forming this loop that you see progressively enlarging and sort of reeling DNA. The DNA is being reeled into this loop. So that's basically the idea of this experiment. So it showed that indeed there is loop extrusion. So with that, so this is the end of 2019, right before the pandemic hits. And now the question is, okay, so that's kind of cool that genome is folded by motors, but what's the use of this? So does it really help in terms of function of the genome? And in many cases, the function of the genome is to facilitate contacts between regulatory regions of DNA and the genes. So on the upper left, I'm showing a particular example arguing that, so this paper from Stefan Mundler's lab, and what this paper have shown is that regulatory regions located in one of these domains need to talk to genes in the same domain, and that leads to normal development. However, if a boundary of a domain is destroyed or deleted, then this regulatory elements start talking to genes in the other domain, and that leads to all sorts of developmental disorders. And there are similar examples in cancer, but basically the take-home message is that these domains that we were talking about, they establish the range of activity of regulatory elements. Somehow stuff in one region, in one domain, can touch the content of this domain, but really cannot touch genes in the other domain. And that's surprising. And it's particularly surprising because if you look at high C maps, you don't really see these domains to be completely isolated. You see maybe twofold difference. So the contact frequency, according to these maps, to experimental maps, is maybe twice higher within a domain than between domains. But they don't really insulate. And there are many misleading illustrations in the literature where people basically replace these parts with the white squares and saying, okay, there are no contacts between that. But it's not really the case. There are plenty of contacts between neighboring domains. And the question is, how does it work? Why we don't see these developmental disorders all over the place? So there are lots of interactions across TED borders. And people have seen this by imaging, by single-cell high-C, by many different methods. So do TEDs really work? That's the question. And I will propose a couple of ideas in this direction in the next 5 minutes, and then we'll try to get your feedback on that. The first idea is that we're really not seeing the whole picture with high-C. Because what we would love to understand are real contacts. And the contact implies that the molecular complex sitting at a regulatory region really touches a gene in the other region. And these molecular complexes are small. So they are maybe 5 nanometers in size, maybe 10 nanometers. So we would love to capture this kind of functional contacts. Instead, what all of these methods, microscopy and high-C methods, are giving us a proximity. And typical contact radii they are capturing are about 100 to 150 nanometers. So instead of seeing this, we're seeing the whole thing. We're seeing everything around. Now we ask, so how much a trouble is this, or would this change the whole picture? So what we've done, we simulated high-C, and we simulated, so in our simulations we use 2 contact radii. We use the contact radius similar to high-C, and we get high-C looking maps. And then we get a contact radius of 50 nanometers that corresponds to kind of non-existing experimental technique that we refer to nano-C. So I'd ask, so are nano-C maps look any different than high-C maps? And the answer is yes, they're much sharper. Okay, the question is, can this help us with this weak insulation of TEDs? Are TEDs stronger insulated in nano-C? And the answer is maybe. And one kind of hint in this direction is that there are techniques better than high-C in terms of their spatial resolution. There is a method called micro-C developed by Ole Rando and Jörg Decker's labs. And in micro-C, the contact radiuses might be somewhere close to 50 nanometers. And indeed, insulation gets stronger. All the features get stronger. So the numbers that are shown here next to these maps are just showing you the contrast of these features. So the insulation, instead of 2-fold, becomes 3-fold. And indeed, in simulations with 50 nanometers, it also becomes close to 3-fold. And I would say, okay, now if we go into 15 nanometers, would it get much stronger than that? And the answer is no. It would still stay within maybe threefold. So it does. So in other words, small contact radius is one idea. So we're not seeing the whole picture, but this idea doesn't really help us completely see strong insulation between paths. So it's not a full. It's not an answer, but it's a step in this direction. But we should remember that sort of these maps are not telling us the full picture. So the second idea is the following. The second idea is that maybe what this mechanism exists for is not to insulate, but to facilitate. In other words, what we're saying is that consider a gene located close to a barrier, to a CTCF barrier. Now consider a regulatory element that needs to touch this gene. Now these loop extruders will be landing uniformly between them. This region is, this distance is say half a megabase. So when one of these loop extruders extrudes and reaches a stop, it would get stopped here, but it would continue reeling DNA from the left into this loop, essentially allowing the gene and the CTCF to scan this whole region for their regulatory regions. And this way, the regulatory region would eventually get to the gene through this process of scanning. And we say, okay, so how would it look like in high C? And I would say in high C, that would look like a stripe at the border of a TED. And we've seen the stripe since 2015, didn't really pay much attention to that. So maybe the stripes are incredibly important because we also see them in many cases in the real maps close to genes. So a gene would be here and then there is a stripe. A gene is Sonic Hedgehog locus. A gene is here and there is a whole stripe next to it in high C. Even in high C, meaning that in micro C and in fictitious nano C, this should be even stronger. And indeed, in simulations, we see about fourfold facilitation. So if we look now in micro C, we see about 5 to 10-fold facilitation. In our fictitious nano C, real molecular contacts or approximate molecular contacts, we see almost 40-fold facilitation at the stripes. So maybe this kind of idea of scanning indeed can increase contact frequency by a factor of 40-fold. And the biological idea is that this CTCFs, these boundary elements, are actually activators. Their main role might be to activate rather than to insulate. And again, in the data, we see many examples of this facilitation at stripes at narrow range. I showed you that already. And the third idea, and I will wrap up here. The third idea is that this mechanism will work even better if the element to be found contains another weak barrier, another weak CTCF site. Again, the scanning would start, the scanning would proceed, but the first element that does not have the second stop sign would get essentially scanned and would continue being reeled into the loop. So it would touch the gene but would not stay next to the gene. The second element that has a weak barrier would stay next to the gene for much longer. So this way you can facilitate these contacts for much more prolonged period of time. So the idea is that, okay, so it essentially becomes a whole system with initiation of scanning, docking, scanning and targeting. And again, in simulations, you can achieve another 5-fold enrichment by placing very, very weak boundaries next to other elements. So I'm showing you again simulations of in nano C. In high C, this would be barely visible. And indeed, in high C, if I take high C, you don't really see any additional peaks along the stripe. In micro C, you start seeing these peaks. And interestingly enough, these peaks in micro C neatly correspond to what's called DHSs, DNA hypersensitive sites. So in other words, regulatory regions of DNA that are accessible to restriction enzymes. So some of them do have CTCF, some of them might be able to stop cohesion themselves. And so it looks like there are stop signs along the scanning path. And again, there are some examples where you don't really see much in high C. You see some signal in micro C of the stopping dots. Another example like that. And this provides another 5 to maybe 10-fold enhancement of contact frequency. What evidence do we have of that? And so one evidence is it looks like, if I look where CTCF is located in the genome, CTCF, and I would like you to focus on the bold lines, CTCF typically has other CTCFs within the first kilobase, so that's the blue line. And it also has lots of enhancers and promoters nearby. In the first kilobase, there are clusters of either enhancers or promoters next to CTCF. And the same is even true for weak CTCF sites. Now, if you look at the dotted lines, so this is again enrichment of enhancers and promoters next to weak CTCF sites. So it looks like CTCFs play a role of essentially indicating where regulatory regions are in the genome to the scanning machinery. That's another evidence that we see these dots generally in the direction of CTCF. What I didn't tell you, again, people more interested in this field know that CTCF is a directional barrier. So again, it facilitates scanning only in one direction. And so by virtue of being a directional barrier, it actually creates stripes in one direction, and the stripes contain dots with nearby regulatory regions only in this direction. So it's an interesting communication machinery along the genome where you initiate scanning at one place, you stop scanning to other places. You can think about this as a transmit of information on one side and receive of multiple receivers on the other side. So overall, what this, sorry, supposed to go forward. Overall, what we're trying to do is we're saying, so we may want to replace our picture of isolated neighborhoods separated by walls with a picture where scanning with barriers allows long-range communication along the genome with facilitation playing maybe bigger role than insulation. But generally, the genome becomes a one-directional track over which there is this process of scanning that brings things together. So it's essentially a genomic communication system. So that's the provocative idea that I want to throw at you in this short talk. And I will thank all the people who did the work. This is my group and our fantastic collaborators. And the people who were focused on this project are Maxim Akayev and Nizar Abdinur in my group, and Jeff Udenberg, who is now a faculty at the University of Southern California. And Samir Abraham helped us with the analysis, did the analysis of the micro-C data. And thank you for attention.
Hey, thank you, Leonid. So now we have time for some questions. I'm going to start off the question round, and I have just a basic question. How universal are these mechanisms? Does it matter what genome you actually use?
It's a great question. So it looks like this machinery of of proteins that extrude loops is present in all organisms. In bacteria, and again, Chase worked on the role of loop extrusion in bacteria. In bacteria, it plays a particular role, maybe also of compacting DNA and of extruding, kind of aligning two chromatids, or two chromosomal arms of the chromosome, folding chromosome in half. In all animals, the system seems to be present. In yeast and single-cell eukaryotes, it's certainly active. It looks like it's an absolutely universal system. What I did not tell you, and so that's something that I will mention in the tutorial, is the same kind of principle, the same loop extrusion machinery can help compact and segregate chromosomes. So its function to facilitate functional interactions, kind of its role is a hypothesis. A more established role, again established in the last 3 years, is in compacting chromosomes during cell division. But it seems to be an absolutely universal thing across all kingdoms of life.
Okay, great, thank you. Let me read a question from Case Decker. So the question is, so far, most detailed experimental evidence for chromosome structure in cells comes from high-C data. What new information do you expect to arise from high-resolution single-cell real-space microscopy?
Thank you for your question, Case. So what I expect to arise mostly from live cell microscopy is evidence of this active process going on inside cells. Because so far, all the evidence we have, as you pointed out, is high-C. That's on live cells or in vitro experiments. So we need to bridge across this by doing live cell microscopy inside cells. The group of Jan Bruges started doing this in the extracts. That's already a big step forward. But it would be really great if we can visualize the process of loop extrusion in vivo. And so, and again, we kind of, several groups are working on that at the moment, trying to see evidence of loop extrusion, or maybe more narrowly, or more broadly, active processes. Because after all, from the physics point of view, so we're talking about not very far from equilibrium polymer dynamics, and the question is, can we see evidence of non kind of active polymer movement within the nucleus? And that's, I think, something that next year or two will bring us.
Great, thank you. So let me ask our panel. I have questions in the wings, but does our panel questions? Agnese.
So can I ask you something? It's very simple, but proximity, is that directly related to something functional? Like if you're next to each other, you're gonna talk, and if you're slightly less next to each other, you're gonna talk slightly less? Is there a linear relationship or something?
I wish I knew the answer. That's a great question. That's a great question. We don't know this. So the belief in the field, and again, enhancers or regulatory regions that are distal have been discovered more than 30 years ago, actually more than 40 years ago,
right.
In the 80s, okay, 35 years ago. And it's known that regions of the genome that are located very far along the chain influence gene activity. How close they need to come, it's not known. And some people are saying, well, maybe they come sufficiently close, and there are experiments that say, okay, they just wander around, they're not really touching each other, and they're somehow influencing the activity of the gene. For me, this is a bit of a spooky action at a distance, because I don't know how two molecular complexes know that they're next to each other without actually touching. That's why what I'm saying is a hypothesis that things need to touch each other such that they transmit some sort of information from one place to another. How this information is transmitted is another mystery.
But so the thinking is that how far you are on average tells you how probable it is that you eventually touch.
Yes,
Oh,
yes,
okay.
that's right.
That makes sense. Okay.
But some people are saying maybe they don't need to touch.
Okay.
Maybe there is a way of communicating without touching in the solvent.
Okay, let me go to a question from Peter Reintenwolde. What determines the maximum values of the fold changes?
Okay, that's a great question. For insulation, it looks like there is, I didn't show you the slide, but for insulation, it looks like there is a limit. And we tried to estimate this limit analytically, and so far we failed. So in other words, the limit arises from the simple kind of the idea is that for insulation, the way loop extrusion works, it just brings two things together inside the motor. So that creates additional facilitation of contacts, and then insulation would be the lack of this activity. Now, but there are also spatial contacts that loop extrusion has no control over, and so they create the baseline. So basically, the limit comes from the fact that things just touch each other at random. And and all this machinery is completely helpless in limiting or reducing this spatial touching. So that sets the baseline for insulation. For facilitation, it looks like there is no limit, in a sense that, well, there is a limit, obviously. So the limit is set by the speed of extrusion and by the number of loop extruders, because if they bring things together, the question is how frequently do they do that? The turnover of extruders is a few minutes, 5 to 20 minutes. So there is a limit, but at least so far in simulations we've seen facilitation easily 1,000-fold. So which would be again more than enough for any reasonable biological process. So we've rarely seen expression can go 1,000-fold in few exotic cases. But generally, yeah, so this can be achieved. By insulation, it looks like there is a limit just imposed by frequency of spatial touching.
Okay, great, thank you. Let's move on to a question from Isabella Graf. I was wondering whether you think that CTCs play a role for differentiation between cell types.
Yes, it's a great question. So generally, this whole machinery, the way I described it, so loop extrusion plus CTCF, the parts of this machinery play a role in if what we're proposing is true, that this basically machinery plays a role in ability of regulatory regions to trigger certain pathways in the genetic program generally, in particular making differentiation decisions. And so far there were few studies, most prominent by the Mirkenschlager lab, where they've seen that indeed in the absence of cohesin, this system is unable to make the correct developmental decisions. Whether this system is important for the maintenance of the differentiated state is a different question. So that we don't know, and I might be slightly skeptical about that. But the proposal is that the system is important for making decisions for differentiation. CTCF, again, the challenge in understanding this is that once you deplete one of these components, the cell cannot divide. The cell can divide a couple of times without CTCF. So a cell cannot divide without cohesin. So it's very hard to perturb the system without killing the cell. And if you perturb it in a cell that is not dividing, the cell has already made its decision not to divide. It's kind of too late. But so far there were a couple of studies, and so it's an area of very active research trying to understand the role of this machinery in cellular decision making.
Okay, great. Thank you. Let's take one more question before we move on to our next speaker. This question comes from Jenny Harju. Do you think cohesins traversing each other would be important for the scanning mechanism to work? Otherwise, you can just take it from there.
I love this question. I'd love to understand this. So far, we've seen evidence of condensing molecules bypassing each other in bacteria. We don't know whether cohesins can do the same. And I would love to understand this, because my guess is that so far we did not consider cohesin bypassing in these models. Mm-hmm. And that's something that I would love to do and to understand in the next few years. My guess is that that would even make this whole rules of engagement even more complicated. But it is surprising how this very simple system of like loop extruders and barriers can generate such an amazing diversity of interaction patterns. Now that they start bypassing, yeah, hell goes loose.
Okay, great. Let's give a virtual round of applause to Leonid for his great talk and questions. Leonid, I think if you can stop sharing your screen, yes. And Agnese, you can start. And while you're starting, Agnese, I'll introduce you. So our next speaker is Agnese Seminara. She did her PhD in physics on turbulent transport and then switched subject, continent, and language when she moved to Boston. In Boston, she worked in the group of Michael Brenner at the School of Engineering and Applied Sciences at Harvard on the physics of microbial systems. She obtained a Marie Curie International Outgoing Fellowship at Harvard and at the Institut Pasteur with Massimo Vergasola, and in 2012 went back to Harvard as an instructor of applied mathematics. 2013, she started as an independent researcher at the CNRS and moved back to Nice. Her research lies at the interface between physics and biology and merges a theoretical approach with numerical simulations and small-scale experiments. She's particularly interested in locomotion and fascinated by the emergence of cooperation among microorganisms. She's going to talk to us today. Oops, I lost my. She's going to talk to us today on sensory navigation in turbulence. Agnese, thank you for joining us.
Thank you, Greg. Thanks very much to everybody. I'm gonna try to make this live and ask you questions, but I think I'm gonna have to speak for most of the time, so you're gonna have to bear with me. I'm gonna do my best. So the subject of today's talk is sensory navigation, and I showed you here pictures, or actually a little schemes of animals that I'm interested into, and it's just to tell you that I'm really interested in the concepts and in the theory that's behind this, but it's applicable to really diverse systems. So the question is very basic. How do we go from one place to another? And here would be the point where I don't let go of you until you answer me. but unfortunately, you can't really interact much. And unless somebody wants to chip in, I'm gonna have to give you some answers already. And there's basically two very different ways that we can go from A to B. One is somebody tells me, okay, there's a traffic light, turn right, and then at the next stop, you go left, and then pay attention, there is a roadblock there, go right, etc., etc. A list of instructions. This is a fixed, something very fixed that I have to memorize and just implement. It doesn't require much of an interaction with the environment, and it gets me there, typically. On the other hand, there are maps where I have to look at the map, figure out where I am, and figure out where I should go. Okay, and here, the only thing I can do is ask you to vote on what do you think we do. But this is the subject of today's talk is about these two very different paradigms. And before going into what organisms do, let me spend 2 words on the differences, on the pros and cons of these two modalities. So the first one, as I said, the list of instruction, you've always, all of you have experienced this. I do this all the time. If I ask directions to go somewhere, then I really concentrate on memorizing. I don't even have to watch much around me, but it's about memory, okay? And if something happens on the road, something unexpected, there's really nothing you can do. You have to ask again. It's a fixed strategy. It's what's called a fixed strategy, or stereotyped, or habitual. These are all terms that I'll use interchangeably. It's kind of easy computationally in the sense that you don't have to compute anything just to memorize. As I said, the big con is that it cannot deal with uncertainty, okay? On the other hand, there is the map and compass paradigm, and this is what's called a flexible strategy. It's goal-oriented. It's intensive computationally in the sense that you have to keep doing computations. Where am I, where I should go? And also the interesting thing is that it relies on some kind of interaction of you with the environment. And it's a continuous interaction. So basically it's based on sensation, or I'm gonna call this a sensation. So the way that an agent interacts with the environment to extract information that are useful for its goal, okay? So I'm gonna talk about these two paradigms in very different systems, and I'm gonna show you at least 2 more times, I think 2 more times, this little scheme down here. On the right, you have the fixed strategy. It's memory driven, and it doesn't deal with uncertainty. And then as you increase uncertainty, you'd better move to a flexible strategy that's better adapted to it, and it relies on sensation. Okay, so you're gonna see this a couple or more times, and it's the one idea that I'd like to you to take home from this 15 minutes. So this would be a good time for panelists to say, we don't understand this, or to say, explain again, or some kind of complaint.
You want us to complain? You're doing well. So,
okay, great, great.
and you've got a mix of predictions over here. So
yeah, exactly. So before going into something I really care about, a couple of slides of introduction about this idea of this flexible and fixed strategies. This is an experiment I really love to show, and I show it to everybody. I think now a lot of people know about this. It's very old, it's 1949. And at that time, most people voted for a list of instructions as the main way we go from A to B. And this is, I think it's the first experiment that sort of like changed the game for this subject. So they put a rat at this position for a few days. The rat would go into this circular arena, and the only boring thing he could do was taking this arm, go at the end of this kind of like little labyrinth and get food. Okay, they would do this for a few days. And then they would move the rat to a different arena. The start would be the same. The rat would get here, but then suddenly the only arm he knew was blocked. Now, this is another point where I would not let go of you until you tell me, where does the rat go? What does it do? It is here. It explores this one arm. It's blocked. What does it do? 3, 2, 1. Okay, this is the answer. And I'm really sorry to have to tell you this because we always get there in the end. But this is the answer. Okay, and I'll give you 2 seconds to think about why this is the answer. Most rats actually take the arm number 6. And this is a demonstration, beautiful, elegant demonstration based on behavior that rats are able to compute shortcuts. And that's a flexible strategy that requires the rat to have a map somewhere in the brain and be able to understand that, okay, this arena kind of looks different, but food was there, so I'll take the shortcut to get there. So I find this amazing, and I wanted to show this to you. And one more point I want to show you, which is also completely unrelated to what I'm gonna tell you about, is that we now know where in the brain are held information about space. There are maps of space that are held by 2 kinds of neurons. They are in 2 different regions of the brain. One set of neurons, this is the probability to fire of one single neuron, and the black is a trajectory of a rat again. And you can tell that this neuron fires when the rat is in this location in space. These are called place cells, and they are like the you are here cell in your brain. Okay, and then there is another set of cells that are similar in the way they function. They only fire in specific locations in space, but this time a single cell will fire in a lot of different locations in space. And if you do this well, they are arranged in a regular lattice, and this is really beautiful neuroscience. For those of you who don't know this, it was worth Nobel Prizes, and there are really a lot of exciting developments. Okay. So this is to say that we do have the possibility to compute routes and flexibly in our brain. We definitely also do stereotype strategies where we follow some kind of memorized location that we feel are interesting for some reason. I'm gonna show you 2 examples, very different, in which this paradigm arises, and here they go. So the number 1 is the when question. So I gave you a where question, which is the question of where should I go to do something interesting? And now I'll talk about, in the next few minutes, I'll talk about mushrooms. They have a when question, and I'll tell you what's the when question that they have to answer. But let me introduce this smoothly. This other guy, this is a sea anemone, and it also has a when question. I'm not going to talk about it, but it's a really fun, interesting organism too. Okay. Okay, so here, what you see on the left is lots of spores, the 8 discs in each of these 2 cells. Those are spores. They are microscopic, 10 microns in size typically. And you see one of the 2 cells kind of like disappears. That cell is shooting the spores out. Okay, this is a very rapid, violent discharge. We've worked a lot on this problem microscopically to show that it's really well designed biomechanically. Here on the right, you see a different way mushrooms are really optimized and finely tuned in their microscopic dynamics. You see this cloud of spores. These are spores that are ejected all at the same time. And by doing this, they go so much further than they would go if they were in isolation. It's a hydrodynamic effect. Fungi just rock. They are fantastic. They have a lot of mechanisms to do this well, blah, 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 years and years of research. And then you realize that the next step after what I'm showing you is this, right? So what fungi do is they eject the spores to travel in space, and they travel across the globe. A lot of fungi, I shouldn't say all of them, but a lot of fungi have spores that are dispersed by air. So they enter into the atmospheric circulation. So what do they do the whole optimization for if the next step is completely stochastic? Okay, so this is the question. And this is my one question. Actually, the one question will come in just 1 second. This is just a conundrum. I've worked for years on demonstrating they are optimized for discharge, and then I have no idea what will happen to the spores in the air. And this is where we did, actually, when I say we, I mean Daniele, did a lot of simulations of atmospheric circulation. He took a big data set of meteorological data. He let spores start from each of 10 different locations in North America at different times of the day, in different conditions, different seasons. And what we found out is that the intensity of turbulence is what determines whether the spores survive atmospheric transport or not. So this is just to make concrete what I just told you about. I was worried about spores undergoing a stochastic fate, and this is really true. If you measure the probability that spores will survive in the atmosphere, it depends a lot on the intensity of turbulence. So it's dictated by turbulence. Okay, and I'll tell you why, which is pretty simple if you think about it. Low turbulent conditions mean that the atmosphere tends to be stable, and stable means that there is not a lot of vertical motion, okay? And in these conditions, spores might start from a set location on a tree or close to the ground. They might get up just a little bit, but then they quietly sediment. They're heavy, so even if it takes a long time, they sediment down to the soil. And in the soil, they are safe, okay? So they likely survive this travel. It's a short travel. But when turbulence is intense in the atmosphere, it typically means that there is a lot of convection, and convection takes stuff up. There is a lot of vertical motion. And if you take tiny particles from the soil high up in the atmosphere, it takes days for them to sediment down. And days in the atmosphere are deadly. A lot of spores will die in those conditions. Okay, so this, it makes sense. There is a very simple physical explanation. The next question is obviously, can fungi tune liberation of the spores to do this well? Okay, and this is my one question. You have a world in which you have to make a decision, not about where to go, but about when to go. And the world around you is turbulent, so it's full of uncertainty. How can you do? What is your strategy? What can you do? Okay. Okay, here's a simple situation for one of these fungi. It's in this yellow star. It's in Mexico. And what I'm showing you here is on the top is the intensity of turbulence. And these are 14 days, and you can count 14 peaks, okay? And right below, it's the probability of survival. And again, 14 days, 14 peaks. When turbulence is high, I'll probably die. When turbulence is low, I'll probably survive. And high and low are very well correlated with time of the day. Okay, so this is turbulent, but on average, the time it takes for you to come down is pretty reproducible. It only depends on time of the day. So in this case, you can ask yourself how much predictive power is held by time of the day. And you can do this more systematically, but you can tell just even qualitatively that this will work. Time of the day predicts survival. So you just have to have a watch and release your spores at a set time of day every day. The next one is a less lucky situation. It's up here in Canada. And I'm showing you here on the top the intensity of turbulence. You can tell there's much less regularity. And right below, I'm showing you the survival probability. And if you just look one of them independent on the other, you're like, oh, okay, this is going to be hard. But if you then correlate them, you realize that when turbulence is high, the survival is low. So in fact, the same role that time of the day was playing above, now it's played by turbulence. If I am able to sense turbulence and release my spores according to some wise function of turbulence intensity, I'm gonna do well. Okay, and this is the dichotomy that I was talking to you about. Here's the result that quantifies what I just told you about. What you see on the x-axis is a regularity factor. We quantify regularity of these two, of all of these situations by a factor that you can imagine. We just looked at the correlations and we measured the peak in the correlation that comes after 24 hours. And by normalizing that, we got a factor that is 0 when you have something that's entirely uncorrelated over the day. So this is Canada. Actually, Canada is 0.1 or something. And then when you have 1, you have a periodic signal. And here's what the example I was showing you above in Mexico. And what I'm plotting here on the y-axis is how better I can predict survival using by sensing turbulence versus memory. So using the watch, time of the day, okay? And this is quantifies what I already told you about. If you are in a irregular situation, you have to sense the world and do something that's flexible and adaptive. If you are in a very predictable situation, you can also go on automatic pilot and just look at your watch and shoot your spores at some set time of the day. Okay, and here's the first time you see this scheme again after I introduced this to you, as promised. Okay, number 2, and I'm not sure how I'm doing in terms of time.
Let's see. We started I think at 4:05, so you should be wrapping up in a couple of minutes, 2 minutes, if that's possible.
Okay, if I have 1 more minute, it's better. But block me when I'm over time.
No.
I'm gonna skip this. It's evidence that fungi do this, but we are working on this. Okay, the chapter 2 is the chapter where. And I'm gonna introduce you immediately to these experiments my collaborators did at Harvard, where a mouse was trained to learn that if it found the source of an odor, it could drink, okay? And this is late in training where the mouse has learned perfectly the task. And you can tell it goes to the green dot, which marks where the source of the odor is, and then it goes to the blue dot where there's water. And occasionally it gets it wrong. It goes to the wrong port first, and then it goes to the right one. And the way this was designed is there were essentially 2 locations right now, actually 3, although we didn't analyze much this one, but there were 3 locations and they were switched on randomly at each trial, okay? And here's performance. So they learn this task very well. They do this very quickly. And here are 2 other more surprising measures of performance. Late in training, they do this very well, but surprisingly, they get it wrong much more often. These 2 guys almost get it to chance level. And the other thing is that they pause much less. So they seem to act rather than think. Pausing is often correlated to cognitive load. Okay. And so here again is my scheme. This is what we think is going on. Early in training, the mouse has no idea where the source is. And so it has to sense with the nose and follow the nose and do a flexible strategy. And indeed, the trajectories look much more wiggly, and they pause much more because it's computationally intensive, it's flexible, and it depends on the odor, it depends on sensation. But then late in training, what happens is that they eventually learn how to go around in this arena, although they have no sight and no sound, but they eventually learn how to go around this arena, and they learn that there are only 3 locations. And so they go around in circles. This strategy is a fixed strategy, it's stereotyped. The trajectories are always the same. There's no need to think, hence less pauses, and there's no need of sniffing the odor at all. Okay, so again, this scheme as promised, and now the simple prediction you can make is that, all right, after training, if I move these locations, they should go back to follow the nose because they don't know anymore where the sources are. And so this is the prediction, and it's actually right. So we had another group of mice trained on a different set of locations, and then we moved them exactly to these 3 locations, and then we measured how many times they got it wrong and the number of pauses. And both measures came back to the level of early in training, although they were well trained. Okay, now if I have 1 more minute, I'll show you. Actually, I'm going to skip this one.
This one, I'm going to be the bad conference organizer and say 1 minute.
1 minute. Okay, so I'm going to skip this, and I'm just going to go directly to this guy. So it turns out how you do this is highly non-trivial, because to follow your nose, you have to sense a turbulent odor, which is highly stochastic. And for some reason that I couldn't understand and couldn't predict, gradient climbing was enough. Okay. So bummer, but on the other hand, we understood why. And the reason why gradient climbing is enough in this arena is because the arena is small. And in this situation, the signal looks like this. So it's highly fluctuating, okay? But it doesn't look like this yet. This is a very intermittent odor. And it happens on the sides of a plume. And in this situation, this is a much more challenging situation. So I'm gonna leave you with the following idea. Intermittency, which is a hallmark of turbulence, is really what makes olfactory navigation really hard. But here's the idea, you can even leverage intermittency to draw information. You have to switch state of mind, but you can use intermittency to give you information about source location. And this is what we are working with Nicola to understand this. I'm gonna conclude with this movie. So I hope this was 1 minute. It's again, Geyer Lab, who's doing this awesome experiments on octopuses, where you see that to get the prey, octopuses go in one go. They don't really wander around. They accumulate information with their arms that are the brain and the nose at the same time. And with this distributed system, they just infer where the prey is in one go. Okay. Here it is. Sorry, I went over time. I'll acknowledge everybody, and especially the students who did the work. Thank you very much to everybody.
Yay, thank you, Agnese. Thank you. All right, I think we should still take some time for questions. I'm gonna start us off. Spores are strong, so why is it that that staying in the atmosphere is toxic to them? Why do they die?
Yeah, so there are measures, and it seems that it depends a lot on the species. I should say spores is like, if there is any biologist here, they'll kill me because it's millions and millions of species. It's a very cryptic kingdom of life, etc. But the few measures that have been done say that UV radiation is one of the deadliest conditions. And in fact, the spores that survive longer, there are spores that are kind of like designed to go across oceans even, and they seem to do this routinely. And these are pigmented. They tend to be pigmented to protect them from UV damage. There are also indications that other species are, so fluctuations are also a problem. Spores have some favorite range of temperatures and humidities, and if you give them too much of a fluctuation, they'll be unhappy. There's not a lot known about the atmosphere. There's a lot known about how they survive in the soil, but it has been a hassle finding data about survival in the atmosphere. It looks like a lot of species that survive hours in the atmosphere, not days.
Okay, great, thank you. Let me take a question from our from the audience. So this is from David Bruckner. Is the Canadian fungus really comparable to a map and compass?
What?
Is the Canadian fungus really comparable to a map and compass? I'll finish the question.
I didn't get the first part. The Canadian what?
Is the Canadian fungus
Uh-huh.
really comparable to a map and compass?
Oh, sorry, the conidial fungus.
Yeah.
So what I'm saying, so I'm obviously this is a very abstract concept, and you should not take this as a one-to-one relationship. But what is comparable is that in one case, you need to sense turbulence. Whatever is it that you do, whatever the fungus can do, it needs to sense the environment and then shoot the spores when the environment tells it it's right to do it, okay? It's like you have a map and a compass and you are like, you have to watch around you and figure out where you are and then decide where to go according to your sensation. I see that landmark, or I know how to, or I had my GPS, for example, and that is the signal. So the interaction with the environment is the key concept that's really comparable in these two systems.
Okay, great, thank you. There's another question from Peter Reinderwolde. Can spores in Mexico detect when the weather becomes more turbulent and then switch to a sensation strategy, relying less on their clock?
That's a fantastic question. And so I've talked to a lot of people to try and get this, get these answers, but there's very little understanding. So it is possible. There are fungi that are cosmopolitan, so they are used to different geographic location and they survive the entire year. So the situation you're describing is possible. And it is possible, of course, in genetic, in species that are cosmopolitan to have genetic variations that adapt locally. It is also possible for fungi to measure the environment. So they potentially can figure out everything you're describing, but there's no knowledge where they do it. The other thing that's known is that Neurospora, which is one of the model fungi and is a cosmopolitan species, it produces spores according, it's tied to the circadian rhythm, the spore production. So it's unknown why they would produce spores tied to their circadian rhythm, but ours is a hypothesis for why you would do it. Maybe you want to shoot them at some specific time of the day. Time of the day is probably easier to implement, so it might, you know, they might not switch between the two strategies and just take that as a proxy of measuring turbulence. And yes.
Okay, thank you.
Can I ask a quick question? You mostly focused on turbulence. Is it clear that other factors in the atmosphere, like wind speeds, air pressures, air temperatures, and things like this wouldn't be also very relevant to spore survival and things like this? Why Is it only about turbulence?
So it's not only about turbulence, but I'm gonna say that most people have focused in, who worked on this problem, thought about where the spores end up. And in that case, circulation and the whole wind situation is obviously important. If you think about how long it takes for them to get back to the soil, wherever they end up, which is the question we asked, then turbulence is definitely the major player, because vertical winds are really not winds, they are turbulent gusts. There's no mean wind in the vertical direction. And so everything is about turbulence, really, what takes you up and down. There are a lot of things that we didn't take into account, which are the canopy and a lot of other details that will definitely play a role. For example, you could avoid all of the problems that I just told you about by designing your spore dispersion to remain within the canopy, where you probably end up sticking to parts of the canopy and you probably are not exposed to the deadly conditions in the open atmosphere.
So how do fungi sense turbulence? What do they do to detect that?
So I'm not sure that they do, but I kind of am, in the sense that, so there's no proof, but these are the sort of like anecdotal evidence that tell me it's possible that they do. So you observe species that eject spores periodically, but also species that eject spores intermittently. Okay, this is the first piece of information. The second piece of information is that in some cases, spores that are very resistant are only released in high turbulent conditions. These are especially the conidia. The conidia are the asexual spores. And for example, the conidia of these two species I mentioned here are very long-lived, of bipolar mites that I mentioned here is very long-lived, and it's only released at wind speeds that are more than 10 meters per second. If you're asking about the molecular mechanisms, these are not understood. There are potential ways for them to do this. There are sensors of infrared light, so that could be a proxy for temperature, which correlates with atmospheric instability. There are ion channels that open up, they are stretch gated, or there are complexes that are activated by a stretch gated ion channel. And in some species, those are exposed to the air, so they could measure wind. So I think they probably do, but it's something that we're actively working on. I think it's a very good question.
Great, thank you. Keisha, did you have a question?
Yes. So you mentioned, so there's a negative correlation between turbulence and the survival of the spores, but I would imagine that there is some biological advantage on a much longer timescale to having your spores dispersed more widely. Is there some kind of sweet spot for that, timing-wise, do you know?
Absolutely. So I think, so that's where I started from, the point of view I started from. But then with the years, I came to realize that maybe we should think about it in the opposite way. If you want to go far, you have to design spores that are long-lived. So in fact, after doing this work, my focus is on, if I have a kind of a certain target about where I want to go, then I have a certain constraint on how long I have to be able to remain in the atmosphere, and that will shape, so the physics will shape the evolutionary forces on the survival distribution of the spores. I'm gonna also say that going far, so for fungi, the environment is very patchy. So there's clearly, you don't want to fall back on the parent fungus. But once you go beyond the parent fungus, it's not really clear to me why you would go a continent away, or just a state away, or 10 meters away. To me, it looks, there are crops, so the size of the crop might be a really important evolutionary force. But I don't think, I don't have an argument for the where. That's why I thought about the when for fungi.
Great. I'm going to propose that we thank Agnese again for the great talk and questions. Thank you. Yay.
Thank you.
That was very nice. And to thank both of our speakers. And now we will transition to the tutorial. So I think all the panelists will automatically get transported to the tutorial. And thanks everyone for coming for the two talks. Now we move on to Leonid's tutorial.
Okay. And I don't need to do, I'm just staying here, right? I don't need to.
You can just stay where you are. You should be.
We change zooms. Okay.
Yeah.
Okay. I hope my computer works. Give me 1 sec. I will quit Dropbox yet again. My internet seems to be slow. Okay. Let me start tutorial. So 1 sec. Yeah, I will share the screen the way I shared it before. Okay. And I hope you can see my slides. Okay, and you don't see all the Zoom panels on top of them, right?
I mean, it looks fine from my perspective.
Okay. So okay, that's good, because I never know whether all the Zoom menus that are abstracting my view are also abstracting your view.
No,
I hope then.
It's good.
Okay, good. So so through the tutorial, what I'll do, I plan to talk for maybe about 40 minutes and take you through some stories, and so we'll see how far we can get. I kind of wanted to be interactive, so I'll be stopping for questions a few times through the tutorial.
So sorry, just to interrupt right from the beginning, I think for the tutorial side of things, also people should feel welcome to just jump in and ask a question if you don't mind. Is that okay?
Absolutely. Okay. So is this also through YouTube? So they'll be asking questions in the chat, in chat, or what's the?
It is on YouTube. Maybe I don't know how the YouTube questions get funneled up here, but primarily we join the Zoom meeting for interaction.
Okay, excellent. So then maybe one of you can read these questions to me, because it's going to be hard for me to keep an eye on them as I.
Yeah, of course.
We'll do that.
Okay, excellent. So kind of the outline of what we may be able to accomplish. I'll talk a little bit about Hi-C first, just give an overview of this type of data, and then about another mechanism that is acting parallel to all of this loop extrusion things that I told you about. And then, if time allows, we'll go back to loop extrusion and look at some of the interesting physical aspects of that process. So what's the underlying question again? So the question is that DNA is very long. It's compacted about 5-fold linearly by being wrapped around these histone cores, these little beads. And that's what we know on one side of the story. On the other side, we know, again, from the pioneering work of Walter Fleming, is that chromosomes look like this elongated things that, like, we draw them usually kind of iconic X-shaped chromosome pattern, but that's how they look like when they're compacted. When chromosomes are decompacted through the interphase, the most of the life of the cell, so they look like kind of random blobs within this big nucleus. So what you're seeing here in blue is it's a DNA stained inside a mouse nucleus. And in red, you see 2 copies of chromosome 2, and in green, you see 2 copies of chromosome 9. And the lessons, there are a couple of lessons from this right away. The first lesson is that chromosomes are compacted even during interphase. They're not completely open up. Remember, the total length of DNA is 2 meters, so each chromosome is a couple of centimeters. The cross-section of a nucleus is about 10 microns. So in principle, if a chromosome had all the space it wanted, it would open up and fill the whole nucleus. But they're somehow compacted, and that's what's called chromosomal territories. And we have no idea as a field what forms these chromosomal territories, what prevents chromosomes from opening up. This can be just topological interactions between polymer chains. They cannot pass through each other. or there might be some special mechanisms. It's still unknown. But nevertheless, chromosomes are each chromosome is territorial. The second lesson is that two homologous chromosomes, homologous means one came from the mother, another came from the father. So two homologous chromosomes are not really next to each other. This is true for all organisms. This is true for mammals. This is not true for flies. In flies, they're paired. And I'm sure there are all other sort of biological oddities. But generally, they don't need to, in mammals, they're not next to each other. Mothers and fathers copies of the same chromosome. So Hi-C data. So microscopy is catching up, but Hi-C is still the main source of information. Invented by Job Decker while he was a postdoc with Nancy Kleckner at Harvard. A 3C technology, chromosome conformation capture, and then scaled up with high throughput sequencing, became Hi-C. What is this technology? In essence, what it does, and I already told you about it in my short talk, but what kind of biologically what's happening, you add a glue, you add kind of cross-linking agent, and in different techniques, different agents are being used. So this agent kind of hugs or glues everything to everything, including two pieces of DNA that might be coming from far on the same chromosome or might be coming from different chromosomes. Now they're glued. Then you chop them into pieces, and then you biochemically ligate or connect two pieces of DNA with each other. And that produces this very elegant hybrid read. So two things that are far on the chromosome happen to be close in space and are glued together into a single physical DNA molecule. So that's a way of, essentially a molecular way of capturing the fact of their proximity. And now you sequence everything. And when you sequence everything, you sequence this hybrid reads, and each time you sequence a hybrid read, you sequence a little bit of the brown part and a little bit of the green part. And then when you analyze the sequencing data, and sequencing in this case would be like 1 billion reads, you map them on the genome, so you know that the brown one came from here, and the green one came greenish, kind of whatever. A teal one came from over there, and that means that I can increase a count in this matrix by 1 each time I see a read that maps to this position. So this way I fill this matrix with counts of 1s from all the sequencing reads. And most of this data are done for non-synchronized cells, and I will show again, if time allows, I'll show you data for synchronized cells. Typically this is done on a population average. So there are single cell Hi-C, you can do it on a single cell, it's going to be just very pixelated.
Can I ask an elementary question?
Yeah, go ahead, Tom.
I guess, is it correct to understand that the resolution in this technique would be ultimately limited by the density of restriction sites on the DNA?
You're absolutely right. So the resolution is limited by the density of restriction sites. There are two resolutions. There is resolution which is linear resolution, so the size of a pixel in this map, and that's limited by the density of restriction enzyme cuts. That's why nowadays people use restriction enzymes that cut every 4 base, that have 4 base pair, every 200 bases, basically. They don't really cut every 200, more like 500. But now the highest resolution is achieved when using an enzyme that really cuts in between nucleosomes. So there are enzymes that just cut everywhere where there are no yellow things. So then the fundamental resolution becomes just the size of the nucleosome, the yellow barrel. And that's the micro-C.
The micro-C.
So micro-C allows you to achieve the resolution. Yes, with micro-C you achieve the resolution of basically 1 nucleosome.
Okay.
Strictly 1 nucleosome.
I had a related question.
It's different in some other biochemical steps, but yeah, go ahead.
I had a related question, which was either from Hi-C or from micro-C. What do you not get that you would want to have?
Well, as I said in my short talk, so there is another resolution, and that's the resolution of what is being captured, or the resolution of proximity, spatial proximity. And here the spatial proximity that we're capturing is still very large, due to factors that we don't entirely understand. maybe due to the fact that stuff just floats and cross-links to each other as it floats, or ligates as it floats inside the nucleus. These experiments are done inside nuclei. But the contact radius is somewhere around, at the best, is 50 nanometers. And the physical contact between two molecules is 5 nanometers. So I want high C that has a spatial resolution that's much smaller. Okay. Much smaller meaning 10 nanometers or below, or around 10 nanometers. And that would be really great if such a technique could work. It's not even obvious, but what you need to do to prevent stuff from floating, maybe freezing.
Just to expand on that, even if you could get the spatial proximity resolution down and the experiment would run ideally, you might still be missing information, right? Because you have one metric in where you're measuring one aspect of chromosome organization, but how much can you really hope to gain from such a measure?
Well, I think it would certainly help me identify some of kind of tell apart proximity from actual spatial touching. But you're absolutely right. Again, it's a snapshot from the population, and it's essentially an equilibrium. I know contact frequency. It doesn't really tell me anything about the dynamics. In principle, you can do high C as a function of time, but it's still an incomplete picture. That's why I think that's as far as you can push this method in principle. But there is also live cell imaging, and so I'm very much into the idea of using live cell imaging as another type of measurement. I think my general approach is that sort of there are experiments and there is theory, and theory I can come up with many models. So I need experiments to narrow down the space of possible theories. And so the more data I have, the more different types of data I have, the more they constrain the space of possibilities. And that's how I generally think about kind of the pathway of discovery, is that it kind of narrows down what is possible. And so that's why kind of from this method, I would want it to be high resolution in space. Single cell, I analyze single cell high C data. They didn't really get me very excited. There are data that are multi-point, and we as a community still don't know how to analyze them. So there are, because here I capture 2-point interactions, but I can capture multi-point interactions, and there are techniques that do that. And so far, what I've seen is largely limited to kind of saying, oh, these things reproduce high C maps. Kind of you sacrifice the higher order interactions. But generally, as a community, we now have this data for a broad range of organisms, which is great, for a broad range of biological systems and mutants. So another thing that I want generally is, so far the pathway that I described is not hypothesis-driven pathway. So I'm saying basically I can come up with theories and I can narrow them down by constrain them by comparing to the data, but in reality, we also need to test hypotheses, actually do targeted perturbations. And targeted perturbations are great, and sort of, these days we have a lot of data of this type also. But even here, you look at bacteria, you look at Cerevisiae, plant, and mouse and human, so there are lots of different high C maps. They look very different, and we don't also know how to think about this. So here I will just throw at you this absolutely crazy high C data from a synthetic system. So it's a work of Xindan Wang, who engineered the strains to produce, and the strains produce this kind of crazy maps. And I wouldn't tell you more than just say that it looks like, to explain them, we need to invoke ability of loop extruding enzymes to bypass each other. Whether that's the only explanation, I don't know. But at least that was one possible model that can explain this kind of absolutely crazy observations. We have a bioarchive about this. So high C can be crazy, but I will skip what I showed you already, and I will, actually, if you want to look at high C data, a very good source is just to go to highglass.io and look yourself at these maps. And this is basically a browser like a Google map. It was developed by Niels Gillenborg and our group just to basically mimic Google Maps. But so what are the features of high-C maps? I'll just mention some features and show you that these features are vastly different between can be different between cell types. So one feature that I told you about TADS features next to diagonal, but in parallel to this, you can notice that there is a checkerboard pattern. And this checkerboard pattern is called compartments. And to an extent, to the first approximation, you can think about compartments as alternating regions, and I will talk about this in a few slides in details, as alternating regions of two types, A and B. So if there is and B interact more with B, and A interact more with A, and then A and B interact less with each other. So that would immediately give rise to this kind of checkerboards. And then there are TADs that I already mentioned, the structures next to diagonal. Another way to summarize what I just told you is that, so if you look at the map, so this is just a fragment of a map. There are checkerboards, obviously. There are checkerboards everywhere. Checkers are they're rectangular, not square, obviously. And the checkerboards are also seen between chromosomes. So if you look between chromosomes, you would also see these checkerboards. And these are compartments. And then in parallel to that, if you zoom in, you will see the structures next to diagonal, and these are called domains or TADs. And they have their own features, there's dots and stuff like that. So it looks like at least two things that are jumping at you are this thing, checkerboards and domains. But they're quite, again, another conceptual way to think about them is that okay, let me actually skip through this. Let me just show you some examples. So usually when I teach, I just browse these maps on the fly, but in the interest of time, I just cut out some examples. So these are two different cell types. On the left, it's an embryonic stem cell, a human. On the right is a human fibroblast. So the same region in two different cell types may look very different. Not always, but they can look very different. As you see on the right, compartments are much stronger in fibroblasts in this region. In another region, TADs are much stronger in ES cells. In fibroblasts, there is just one big square. You should learn to ignore these white stripes. I completely ignore them. I internalize this knowledge and skill, but sort of these are regions of the genome that are just unmappable, so we just don't know what's happening there. So this is terra incognita because of repeats. We don't have to worry about it. But beyond them, there are lots of structures next to diagonal on the right, on the left, and there is one boring square on the right. Shouldn't necessarily be so. Again, another example in fibroblasts. Lots of structures of compartment type, checkerboards, and the non-checkerboard in ES cell. So these are kind of two descriptors of the data, and so they're different in different cell types. We don't know why. Any questions about the data? I will take maybe quick questions and then start talking content.
So for the different, you showed this previous slide, right? And then they are very different. But then if you do the experiment twice, how similar is
Oh, okay. Oh, they're very reproducible. This data is strikingly reproducible. So actually what I showed you is typically three replicates are being done, 3 biological replicates. You compare across replicates. These features are very, very reproducible. And the dynamic, so you certainly have noise, but so the dynamic range in this data is also striking. It's typically 4 orders of magnitude, 5 orders of magnitude, in the best data set, 6 orders of magnitude from the darkest to the lightest. So this data just completely spoils us because we don't do any statistics on that. We don't do p-values. You just see things.
But even though the whole thing is very reproducible.
The whole thing is very, very reproducible. Meaning, it's very reproducible because it's an ensemble average measurement.
Right.
So once you go into single cells, nothing is reproducible.
Exactly. That's what I guess. That's what I'm trying to put my head around.
Oh yeah.
Supposed to be
yes, absolutely.
Dynamic, and then it's just this is the bulk somehow that we see.
This is every map is averaged over 1 million cells.
Right. I see.
So these are features of the ensemble, not features of individual cells. Once you go into single cell high-C, you get very pixelated maps, and then it's very hard to make sense of it without just averaging over cells back. Kind of it's still you can still see all these features when you start looking for them, but then you need to do statistics, you see enrichments and all of this, all of the usual things stuff. But on these maps you just see things. So let's talk about compartments. It's an interesting story, and I want you to at least want to get you through that. So what are compartments? So compartments, I told you, are these regions of the map that give you checkerboards. They are, you can basically, you can take a high C map and find the best representation of this map as a two-state by two-state model, where you would say there are every region has a state. It's either an A state or a B state. And basically it's the first eigenvector of this matrix. Somewhat corrected matrix, but otherwise just the first eigenvector. And when it's high, it's A. When it's low, it's B. And then B would be enriched and AA would be enriched. And then AB is depleted. So that's, you can model this map as just as a two-state representation of the genome. What's surprising is that this representation of the genome actually makes sense. Typically, A regions are gene-rich and have lots of genomic activity. So this part of the genome is being read by the cell. And B regions are typically either gene-poor or these genes are silenced. So that's a representation. So what do we know about them in imaging? So people have been studying these regions in imaging. So if you take regions that are generally gene poor or silenced, they're called heterochromatin. So this is B, I will be calling them B. And it's known that these regions are typically at the periphery of the nucleus. And regions of the genome that are active are typically in the center of the nucleus. So there is a very nice kind of spatial separation between active stuff in the center and inactive at the periphery. Now the question is, what kind of mechanism can lead to this separation? Again, on the right you see imaging. Again, this is microscopy, so let me explain to you what you see here. Again, you can stain these regions in different colors. B regions are here in red, in contrast to the previous slide, and A is in green. These red blobs are actually particular nuclear organelles, nucleoli, that are also coated with B. Why they're coated with B is not entirely known, but they're somewhere, and they're kind of, for this argument, they're not incredibly important. Generally, you see the rest of the red at the periphery, a very thin layer at the periphery, and then the center is filled with red. And the question is, what kind of process leads to this kind of phase separation of red of A and B?
Can I interrupt with a question? I was curious about.
Yeah, go ahead.
Is the statement that a two-compartment model is a good description of this data, of this matrix? Is that a statement about the spectrum of eigenvalues of the matrix? Or
that's a great question. It is. Basically, what you see is that the first eigenvalue is very large. The second one is noticeable. The third one is there, and then beyond the first three, there is very little signal. So if I start doing this for different experiments on the same cell types, you basically see that the first three are reproducible and the others are not. The first one is A and B. The second one typically shows some signal along the chromosome. Because chromosomes in human, for example, they have centromeres and telomeres, and so the second eigenvector typically decays from centromeres to telomeres, reflecting some other folding of chromosomes. But you're absolutely right. So it's a statement about the spectrum. And now with high resolution data, we start seeing that maybe first 4 or 5 of eigenvalues are noticeable. Yeah.
Great.
But the question is, again, what kind of molecular mechanism leads to this? And it's obviously highly reminiscent of behavior block copolymers. So if you have a polymer that consists of blocks of A and B, and then if A is attracted to A, or B is attracted to B, or both, then it leads to immediate what's called microphase separation. So the difference between microphase separation and phase separation is that in microphase, the sizes of phases depend on the sizes of stretches of these blocks. So if the blocks are short, you get small inclusions of say, A inside B. As blocks get longer, these inclusions change their shape from blobs to rods to layers and so on. So these are classical pictures from polymer textbooks, very well studied and used in industry all over the place. The chances are that what we're seeing inside the nuclei are the same. And so several groups suggested that. They basically say, what if I create a block copolymer, and then the block copolymer would certainly fold by itself into a state that would give you a checkerboard. And that's already kind of conceptually good conceptual advance, but it does not answer the question about molecular mechanism, which is interesting because you want to say, is this that active regions of the genome are attracted to other active regions? Is it AA? Is it BB, attraction of inactive ones? Or there is a third possibility, is that the nucleus here also has a, okay, I want to show it somewhere. Okay, I don't have the real imaging, so I just drew it on top of the slide. The third possibility is that there is a nuclear component called lamina, and this lamina coats the nuclear envelope. And there is an attraction between B and this. There is particular anchoring of B to the lamina. And is it possible that attachment of B to the lamina is a mechanism that would lead to this phase separation? So that's the question that we started with. And what really helped delineating between these three mechanisms is a biological oddity, is a rare biological system. And this biological system are nocturnal mammals. Most of mammals are nocturnal, in fact. We are, and other big apes are an exception. Nocturnal mammals have, in their retina, have rods that are used for seeing in the darkness. And these rods have an architecture of the nucleus that turns them into natural lenses. And this architecture is such that inside, so instead of having B at the periphery, they have B in the center. And this B is so dense in the center that the optical density of the nuclei become high in the center and low at the periphery, so they become lenses. They focus light. And that was discovered by Irina Solovey and her group several years ago, and they keep exploring this. But for us, it was an interesting example where you basically take a nucleus, and instead of getting having a conventional architecture with B at the periphery, you get B in the center. And all that's happening to the cells during development is that they lose lamina. Oh, they don't lose lamina, let me take it back. They lose a way of attaching chromatin to the lamina. So their chromosomes are not attached to the lamina, and their nuclei naturally, over the course of about 6 weeks, invert into this invert architecture. The first lesson to take from that is that it basically means that to separate green from red, you don't need lamina. It's something about intrinsic property of B or A that leads to this inversion. Yeah, this is just conventional nucleus for comparison. These are just other neurons. Other neurons have a thick layer of red at the periphery and the green at the center. And again, this nucleoli that are here and there. So we ask the question, so how can I reproduce this imaging data? What kind of molecular model can reproduce this imaging data? And we've also done Hi-C on them in collaboration with Job Decker. And Hi-C for this inverted nuclei look completely conventional. These are just normal checkerboards, because what the checkerboard reflects is just the separation of A and B. It doesn't really tell you which one is in the center, which one is at the periphery. And this kind of comes back to Chase's comment that, so what do we want to know? Like, ideally, I also want to know where things are in space. So contacts are certainly not the whole picture. So that's a good example where a contact map really doesn't tell me much. It just tells me that things are separated. So what I did not tell you is that in mouse, there is another third state of chromatin called C chromatin, or constitutive heterochromatin. These are regions that are not visible in Hi-C. They're highly repetitive, so these are these white stripes, but they are very condensed, and they are and they have a particular property allowing to map them differently in microscopy. So in microscopy here in imaging, you see them as the pseudo color that's being used as blue. So they form this very, very round blue core, and that's decorated with B, and then the A is in the rest of the periphery of the nucleus. And the question is, what kind of mechanism can give rise to this? And say, okay, that's a block copolymer, fine. C is always at one end of that. That's the structure of mouse chromosomes. Then there are blocks of A and B, and there are three types of chromatin, 6 types of interactions. And the question is, how do they need to interact to give rise to this kind of pattern? So we tried all possible 6 relative strengths of this elements. You can choose once you set BB and CC, for example, you can choose BC such that they are not going to mix. You need to have BC lower than the arithmetic mean of BB and CC. So there is an old kind of classical result in polymer physics how strong this should be, such that the regions demix. So you make sure that they demix, but there are still your space of models. So this comes back to kind of philosophy of how we approach this. The space of models is huge. I have 720 classes of models. So, but I can enumerate them without kind of on a computer and see what kind of inequilibrium, what kind of pictures they can give rise to. And what I see is that, so these are simulations, they give rise to all sorts of patterns. And I'm showing you the first 8, the best 8 that agree the best with imaging, and we quantify comparison between this and imaging data. Image microscopy is above, by the just radial positions of red, green, and blue. So the best 8 here at the bottom of my plot, so model number versus distance to the experiment. So they provide the best agreement, and all of this one, so my measure kind of comparison to experiment worked. Blue in the center, red at the periphery, green, red next to blue, and then green at the periphery. But you can get all sorts of crazy structures. So these are this crazy Mickey Mouse, mushrooms, also kind of a zoo of different things, depending on the relative strengths of this 6 types of monomers. 3 types of monomers, 6 types of interactions. So we say, okay, what's common about this 8 that reproduce the experiment? And it turned out that for all of these 8 best models, they have the strongest attraction is CC. The next one is BB, and BC should be in the middle, as I told you. Kind of we can set it up such that there is a nice phase separation. And then all the interactions of A are very small and actually can be set to 0 without any loss of the phenomenon. So basically what it tells me is that attraction between C and B should be strong, attraction of A should be none. And that would give rise to the inverted nucleus. And then one side, and that's cool. And that I can also fine-tune this parameter such that I can reproduce high C. High C provides some additional constraints on this. And then I can reproduce the conventional nucleus by just adding the lamina. I add back this gray layer at the periphery of the nucleus, and I add interactions of B to the lamina and C to the lamina, and again also can try to find in the space of these 2 parameters where the good agreement is achieved, and basically B should be attracted to lamina as strongly as B is attracted to other B, and C should be basically irreversibly tethered to the lamina. So what did we learn from this? We learned that A interactions are completely dispensable for this. That this major phase separation in the nucleus, or microphase separation, is driven entirely by interactions between heterochromatin, B or C, still heterochromatin, inactive regions of the genome, or basically junk DNA, to the first approximation. That's what's important for compartmentalization, for phase separation, and then attachment to the lamina is needed for positioning stuff to the periphery. So without it, nuclei would be inverted. So basically, the default state of a nucleus is to be inverted. The question is why all the nuclei are not inverted, and we have no idea. The default state should be to be inverted. Invert is new conventional.
And I have a question.
And then on top, yeah.
Yeah, this is all, of course, for eukaryotic chrome for cells and the nuclei. Is there any implication for bacteria of this?
Great question, Case. No. Because in bacteria, genes are relatively uniformly dense, and there are no compartmentalization. There might be some gaps. There are other structures in bacteria, but they don't seem to be checkerboarding. Not even in yeast.
Okay.
Even in yeast, there are no checkerboards. This is okay. In fact, I can tell you something that kind of these checkerboards emerged in evolution, not even for all animals. There are some very early animals that do not have this.
Is that related to chromosome size as well? Genome size?
Maybe it's a great question. It looks like it might be. It's very hard to delineate this from other early evolutionary inventions, but it might be related to size. Indeed, very early animals have relatively short genomes and very dense gene organization, like yeast, basically.
Yeah.
And then So this is like for really grown-ups, like with very long genomes and sort of uneven distribution of genes, right? But I don't know. Actually, great question is I don't know a single. I know only one cell type where there are no compartments, and the cell type is maternal pronucleus. So oocyte has compartments. After fertilization, the nucleus that comes from the sperm lives separately from the nucleus that stays in the oocyte. And at that moment, oocyte nucleus, maternal nucleus, loses compartmentalization at the moment of fertilization. Paternal one has compartments. They're weak, but they're noticeable. In maternal, it looks like there are none, or so weak that we can detect them. And then as the embryo grows, compartments get stronger and stronger. And so there's only one cell type without compartments. But it looks like go ahead.
Yeah. So you described these interactions between A, B, and C. So what are these interactions? What should we map them to on the DNA?
Great question. I don't think anybody really knows this. There was a hypothesis that BB attraction is mediated by a particular protein that recognizes B regions, and I'll tell you what's special about B regions in a second. This protein was called HP1, and there are several studies, published and unpublished, where people removed HP1 and there was no change in compartmentalization. It's certainly not HP1 itself. It might be direct interactions between B regions. Okay, so what's special about B regions? B and A regions carry different chemical modifications of histones. Histones are these yellow proteins that are yellow in one of my earlier slides. Some of these proteins that are binding DNA and forming nucleosomes, so they're chemically modified. In active regions, they carry a particular set of marks. In inactive regions, they carry different marks. They're largely methylated. So maybe it's the methylation of histones that makes histones stick to each other. There are works supporting this idea. There are works from Daniel Gerlich's lab in Vienna. There are other studies suggesting maybe these are actual interactions between modified histones. Other people are suggesting it might be mediated by a particular protein, but the actual molecular player is still unknown. Okay, so I will start asking questions already about, so I can take maybe another question about this and then we'll show you something else and it will be, or is it a good point to maybe just take questions? I don't know, you tell me. I
have a question exposing probably my ignorance. Maybe it's a stupid question, but you started this tutorial with a beautiful picture of the chromosomal territories. And in this compartment picture now, you have the whole nucleus filled with this stain. So is it correct to understand that that's a mixture of all the different chromosomes that still have territories?
It's a great question, Tom. It's a fantastic question. It basically means that each chromosome somehow orients itself in such a way that its B part is at the periphery and A part in the center. It's like a dipole, sort of. Yes, it looks like chromosomes are doing that.
The territory still exists, even
yes,
in that condition.
Even in individual chromosomes, right? So this is something that's not entirely understood. So what do chromosomes do in this respect? And there were beautiful imaging studies that came out a few months ago, showing that indeed chromosomes are sort of like dipoles themselves. They have B at the periphery and A in the center, but I can tell you from our work, it looked like maybe chromosomes are coated, kind of a little inverted nuclei themselves. They have B on one side, but then they have largely A at the periphery and B in the core. So how to combine this block copolymer picture with chromosomal territories is not entirely obvious to me either.
Yeah, that's interesting. Can Hi-C sort of potentially map out interactions between chromosomes? Is that?
Yes, absolutely. No, Hi-C absolutely maps interactions between chromosomes. Sorry, I didn't. Yeah, you see interactions between chromosomes in Hi-C. In fact, most of the map is these interactions between chromosomes, and we see beautiful compartmentalization there. In fact, this checkerboard is absolutely visible between chromosomes. But kind of what's happening inside each chromosome in terms of spatial positioning, we don't quite know, and that's why imaging comes and helps us.
Yeah, we have a question from the chat by Youyang Lu. How do you explain that compartments can still be maintained after depletion of lamina?
Okay. So that's basically the whole notion here. I will go 2 slides back. Is that what's important for compartmentalization is that it's not the laminar attraction, but it's the BB attraction. And attraction to the lamina is only needed for compartments for B to stay at the periphery. Without lamina, B will gradually over 2 weeks will go to the center. I may or may not have this slide. If you give me a second, I'll tell you. I may have a slide to show this gradual process. No, not in this deck. Okay. There is a gradual process of inversion. So lamina is dispensable for compartmentalization. It's not needed. It's all glued by BB.
Sorry, I just saw Keisha. Did you have a question? I couldn't.
Yes, but I was waiting to see if anyone else had one before me. But yeah, so my question is actually possibly quite naive. This is not something I know that much about. But how much is known about the dynamics here? So how much of everything is jiggling around? Because I would imagine that for the Hi-C system, for the cross-linking, rather than just proximity, what you're actually looking at is perhaps regions which are simply less dynamic, right? And then what you're actually in the end mapping, maybe your attraction energy or your sticking is looking more at like regions which are moving less or moving more in the case of like a lower sticking energy. Could that be a possible interpretation?
That's an incredibly deep question, I can tell you. You're absolutely right. So stuff does move. Typically, Hi-C experiments are done sufficiently quickly. Okay, it depends very much on the protocol. So in the best Hi-C experiments, they add formaldehyde, which glues everything relatively quickly to everything else. And then you wait for half an hour and you do the rest of the experiment. In some cases, people add formaldehyde and wait overnight. So stuff can move. In normal cells, without the glue, the typical number for the displacement is about 1 micron per day. So the nucleus is about 10 microns. So it's 1/10 of this in a day between cell divisions. So stuff is not incredibly mobile at this whole nucleus scale. And the effect of mobility on Hi-C is not entirely understood. So what typically when we analyze this data, worry about is that everything got glued. That's great. Then you chop everything in pieces. At that moment, stuff starts floating. So when it floats, that's when you get all sorts of additional interactions that were not really present in the intact cell. Job Dekker's lab did a beautiful experiment where they've done the following. They took a cell, did not add a glue. They cut chromosomes into pieces, literally chopped them into fragments of a few thousand bases, and waited for 2 hours and did high C. And it looked like a regional high C.
Okay.
Then they waited for 4 hours, for 8 hours, and stuff did change, but very gradually. And then they waited for a month, and you can still see compartments. So they completely liquefied the nucleus. They chop it into pieces and equilibrated it for a month. And you can still see compartments. You lose some other structures, you lose TADs, you lose lots of other features. But there are still some features that survive for a month. And we're still trying to understand this. So we don't have a complete physical picture of what's going on. These interactions are somehow preventing stuff from floating away. We don't know.
Mm-hmm.
So does this imply this is very gel-like liquid-liquid phase separation then?
Yes, it implies that there is a good gelish component, I would say. Yes, that some stuff does not flow away for a very long period of time. Again, everything gets, I'm saying it's something still visible in high C maps. It doesn't mean that things do not change for 2 hours, but things do start to change at 4 hours and 8 hours, so it floats. But indeed it floats slowly. So there is a good gel type component, and I wonder whether lamina may be playing a role here. So I think your plans to kind of get rid of the lamina and see what's happening there. It's not an easy experiment, sort of, and the inverted nuclei can only be seriously done in the primary tissue. So these are not cells. This is a whole. This is a mouse. But yeah. So there is a good degree of gelation in the system. Okay, tell me, so do you want me to tell you something else, or is this a good time to take more questions? What's the...
I think people are not shy about asking questions, so you can just keep going.
Okay, sounds good. Okay. So
I'm going to jump in because there's a little follow-up I wanted to make on the discussion on the compartmentalization, because I remember this hypothesis that the heterochromatin is slightly stiffer than the euchromatin, and this by itself could be a entropic driving force for getting the heterochromatin to the periphery. And that would work on the level of individual chromosomes, but also collectively.
It's actually a great question. My computer is
I'm not sure it's a
slowing down,
question,
which I don't like.
It's in the...
Yeah. No. That's true. The question, kind of, to me, the question is this enough to achieve the degree of quantitatively strong compartmentalization? For example, there was a proposal from Joanny and Grossberg that sort of it might be a non-equilibrium system with a differential temperature, with high temperature in A and low temperature in B, and that never got us compartments strong enough to see in the system. So whether differential stiffness would be sufficient, yeah. So in principle, stiffness would make things more flexible in the center, more rigid to the periphery. But whether this is sufficient to get quantitative.
We thought we were going to try this out, but we didn't. So I
need to turn off my Dropbox in 1 second, because otherwise the computer is freezing and it's not good. This computer is very old. Okay, let's okay, I cannot turn off the Dropbox either. Unless I force quit. Give me 1 sec. Because it slows things down considerably. Okay, hopefully that would make things faster. Okay, so coming back to loop extrusion. So that's as far as the global phase separation. So the level of loop extrusion, again, I told you about the problem in forming these domains. And the fundamental problem, why do we need loop extrusion, is that what became evident about this small, remember in my short talk I mentioned the small squares, So what's important about the squares or domains is that they're formed because there is a binding of a protein somewhere at the boundary of these domains. And this is really hard to understand in terms of scales, because it sounds like a binding of a relatively small CTCF protein somewhere in this jumble of DNA controls how frequently two blue regions that are separated by hundreds of nanometers inside the nucleus interact with each other. And that to explain this, we need loop extrusion. We need some active process that kind of locally compacts chromatin and senses this barrier, and that's why we had to invoke this hypothesis of loop extrusion. So what are these loop extruders? Because since I spoke about these domains earlier, so what are these loop extruders? So these extruders are complexes of the SMC family, and they look like other molecular motors. They look strikingly like other motors because they have an ATPase. They have an ATPase heads that, when together, they form an ATPase domain. They form a hinge, and they have long coiled coils that are very flexible. And again, cases the group just showed that these coils are incredibly flexible. In other molecular motors, they're more stiff. They also have two ATPase domains, and they also have something like a hinge or something else at the other end of the myosin or kinesin. So these guys, other motors, can walk on their substrates. So the question is, how can loop extruders extrude loops? These are proteins of the SMC family, and I will not give you an answer to this. We don't know. The answer is we don't know. But what we do know is that they're also helping not only solve the problem of creating domains, but they also help in compacting chromosomes. And again, sort of just to remind you, so during interphase, the nucleus consists of these individual chromosomes. During metaphase, before cell division, each chromosome gets compacted into an elongated structure. Two sister chromatids are separated, next to each other, but separated. And these are these chromosomes that end up being in separate daughter cells. And again, I will tell you about this process in 1 second. So thinking about this with John Marco, we say, okay, so what's a good way of describing the system, even without barriers, just activity of loop extruders? So if you take loop extruders and so there are two length scales in the system. There is a size of a loop that an extruder forms before it falls off the DNA, and there is a separation between loop extruders. So these are the only two length scales in the system if you say that the total length of DNA is much longer than lambda and D. And that immediately gives you two regimes. When lambda is much smaller than D, then loops are independent. They don't really sense each other. That gives you a sparse regime that might be more relevant in the interface. When lambda is much greater than D, then you get a very dense regime when you have loops within loops. So loop extruders start landing within the pre-existing loops because they're turning over. And that creates very stable loops such that individual extruders exchange, but the loop stays. And for this system, we solved for the in steady state. You can write down the death rate and the birth rate of the division rate of a loop and the death rate of a loop, equate them, and this gives you a way of estimating the average loop size, which is approximately D log lambda over D. What's important about the dense regime is that all the DNA is in the loops. There are no gaps, because they will continue extruding until they meet each other. In this little simulation, you will see what's happening in the dense regime. This is a 1D simulation. So these are loops here shown as arches. Zoom may not be happy about this. Okay, let's optimize for screen. Let me go one slide back. Okay. No, back, not forward. Okay, I hope you can see this as an animation. So these are loops growing, and so all the DNA ends up being in the loops, and then the loops start forming inside existing loops, and the numbers show how many extruders are at the base of each loop. And then there is a steady state in the system with a characteristic loop size. So in 3D, this process really is amazing, because what it does, it takes a randomly crumpled polymer and it converts it into something that looks like a chromosome. So what do we know about chromosomes? An interesting feature of chromosomes, and it's not entirely known why this feature is important in mitosis, is that if I take a polymer, a chromosome, and I paint it in a rainbow fashion from red to orange to yellow, then regions that are close along the chain end up being close along the chromosomal body. So this cartoon just explains how chromosomes should look like. So colors should be separated such that consecutive regions are organized linearly along the chromosome body. The second puzzling thing about chromosomes in mitosis is that they're compacted into an elongated structure. Which is surprising, because if you just add some cross-linking agent, it would try to minimize the surface area and would compact the chromosome into a ball. Instead of a ball, you get an elongated thing. And the third property is that there is an axis. Each chromatid gets its axis, which is visible here. And this axis is formed by SMC proteins called condensin, which is also a loop extrude, as again, Case has shown. So that was the hypothesis, that maybe this can be achieved by loop extrusion, and oops, it kind of goes back and forth. And indeed, so what I'm showing you here are simulations of this process done by Anton Golobrodko, who is now a PI in Vienna. And what you see is that loop extrusion, if you see this animation, it kind of gets slow on my side. In the simulations, loop extrusion acts on the polymer. And on the left, you see it's colored according to its position along the chain. On the right, you see it's transparent, but you see individual loop extruders. And what you should be able to see is that colors get separated, the shape of the chromosome gets kind of elongated, and the scaffold is being formed all at once. In addition to loop extrusion, the only process that you need to assume that the chains can pass through each other occasionally. This in real life is facilitated by another enzyme called topoisomerase 2. So that's a remarkable property of loop extrusion process, to essentially compact stuff into elongated things with scaffolds. And moreover, if you take two polymers, so that's another problem. If you have two chromosomes, two sister chromatids, so they got replicated. You have DNA, DNA got replicated. Now one copy should go to one daughter cell, another should go to another daughter cell, but they're originally completely intertwined. So if you start with two polymers, red and blue, that are mixed, and now subject them to loop extrusion, what you'll see is that gradually, as the simulations run, if it runs, it doesn't look like it wants to run. Okay, this is Case's experiment. Too early for that. Okay, so let me scroll this. I don't know, it doesn't scroll it forward. So what you should have seen in the simulations, if they worked through Zoom, is that the two polymers start separating from each other as a result of loop extrusion, and gradually red and blue get separated. So you get two chromatids, and these chromatids are now get separated. And this is entirely through the activity of loop extruding enzymes.
Can I ask a question about this?
Let me exactly ask, so I have a designed pause for questions here. Yes, go ahead.
To what extent is this mechanism compatible with the number of condensins that you might have in a cell and the rate at which they're extruding loops? Is this really a feasible mechanism through which you can?
Yeah, that's a great question. So we're worried about that. Basically, it depends on the loop sizes, and if we have a few more minutes, I can show you high-C data for this process as a function of time. So the loop sizes that you get are about 100. So okay, it's complicated, but the smallest loops are about 100 kilobases. The process takes about half an hour. So it is compatible with the speed of loop extrusion, but it would basically so it would kind of, because right now we kind of know that loop extrusion proceeds at a speed of about 1 kilobase per second. That's what Case Decker's experiments are showing in vitro on naked DNA, but there are similar data for bacteria, showed similar speed in bacteria, in live bacteria, and there are some indirect evidence of a similar speed in mammalian systems. 1 kilobase per second, you certainly have 100 seconds to form 100 kb loops. Because typically prophase takes about 10 minutes. So what I showed right now is the compaction to prophase, and that takes about 10 minutes. So it is comparable. So in terms of number of molecules, you basically need 1 condensate per 100 kb. And you do have enough of them. So there are direct measurements of condensates done by Jan Ellenberg's group and Christian Herring's groups together, and they did show that you have 1 condensate every 50 kb. So there are enough molecules, and there is enough speed. Another interesting question is whether there is enough energy for this, because they consume ATP. And especially, again, compaction takes only 10 minutes. You can spend lots of energy at this moment. What I argued before is that this process is ongoing all the time through the life of a cell. And we estimated the ATP budget of all of the cohesins extruding loops all the time. And it turned out to be a tiny fraction of the total budget. I was joking that it's smaller than the science budget in the United States compared to the total GDP.
You also can mention that the motors, that these SMCs are like extremely efficient motors, because they use only 1 ATP for hundreds of bases. It's very, very different from kinesin, myosin, and all these other motors. Very different class of motors. Really. Very low ATP.
Exactly. They're extremely economical. Yeah, like scientists. Yeah, but the total budget is relatively small. So speed's budget, that is something that I think we understand, but there are certainly certain things about mitotic compaction that we don't understand. There are some physical questions that are completely perplexing, despite of all of the there are certain problems that cannot be solved by loop extrusion, or at least we don't know how, and I can tell you about them. So. Okay, if there are no more questions, what I'll do, I'll jump to actually, I want to show you this temporal high C data, because I will not show you Case's beautiful experiment on yeast condensate and the question about the mechanism, but I will jump into this part, which I rarely actually present because there is no time for that. So this is, my computer is getting slow and it's kind of makes me worry. There's are 2 papers which go Decker's lab, and the last one is in collaboration with Bill Earnshaw. So what you see here is microscopy for tightly synchronized chicken cells. These are 2 collaborators in the chicken here on the slide. So these are chicken cells, and after 0 minutes, they're still in the interface. 2.5 minutes, the chromosomes start to compact. 5 minutes, you already start seeing individual chromosomes. 7 minutes, there are clearly individual chromosomes. 10, they're already lawn and sausage looking like what I showed you in the simulations. And then 15, 30, and so on, they get shorter. They get shorter, they get more compact. And the puzzle, there is a physical puzzle here. As I'll argue, to get this kind of structures, you need loops to be collapsed. You need this polymer to be in a poor solvent condition, so it needs to collapse. But for collapsed polymer, but if the polymer is collapsed, then these chromosomes they will start sticking to each other, because you can achieve collapse of a polymer if monomers attract each other. So if monomers attract each other, they would form this kind of structures on top of what I showed you, but then chromosomes would be sticking to each other. And as you see here, beautifully, chromosomes do not stick to each other. And we don't quite understand what makes chromosomes collapsed and not sticking to each other. We understand what gives them the shape, loop extrusion gives them the shape, but we don't understand something about this microscopic collapse. In any case, so you could do high C as a function of time. So this is high C at 0 seconds. You may be able to see the checkerboard and the structures next to diagonal. At 2.5 minutes, the structures are still there, and you see chromosomes still largely decondensed. At 5 minutes, chromosomes become visibly condensed, slightly condensed on the right, and these features start getting weaker. At 7.5 minutes, chromosomes are already individualized, and stuff along high C, you see all these interface features vanishing. 10 minutes, very tightly already kind of long chromosomes. 15 minutes, 30 minutes, chromosomes are neatly packed. What's also interesting, and that's another puzzle, is that what I see on the left is a high C that lost all its features. It's just a decay, just intensity changing from as I go away from the diagonal. But what's also puzzling is that I'm getting a second band. And the second band basically tells me, if I go back through the series, there was no second band, 10 minutes, no second band. At 15 minutes, I start seeing second band. 30 minutes, the second band moved further from the diagonal, meaning that the chromosomes are spiraling. And I don't think I have data for 60 minutes. Oh, I do, okay. So the second band is still visible after 60 minutes. So chromosomes are very tight, but it looks like they're spiraling. In fact, the only way to explain this data is to invoke a spiral. So all this data can be collapsed in the form of curves, which is the contact intensity. Contact density is a function of distance from the diagonal. So this is in the first 10 minutes, so no spiral. And to explain that, we developed an analytical model where you have loops emanating from the scaffold. And it turned out, so again, this is the work of Anton Kolobroidko, who is now a group leader at IMPA. And it turned out that you can explain this by an analytical model very nicely. So most of the curve can be explained analytically by loops that are emanating from the central scaffold. And the loops needs to be correlated in their positions. What makes them correlate in positions, we don't know. And then beyond that, we say to explain the spiral, so the data here in magenta. So we can explain the spiral, but to explain the spiral, we need a model where the loops are forming a spiral staircase. And what makes them go spiral, we don't know. Again, loop exclusion just gives you a series of loops, gives you an underlying structure, but the internal organization inside the structure is completely mysterious to us. Oops. Okay, my computer got even slower. Okay, that's a bad sign. I think my slides are frozen already. So as long as my video is not frozen, I'll take questions, but I'm afraid so I can't make it run any further than that. It's a good stopping point. So if my video disconnects, I will reconnect in a second, but it may happen.
So could the
spiral... okay, it did go forward. Okay, good. Good. Yeah, please ask your question. I will just show a slide.
Sorry, could the spiral shape... So it's a lot more regular than just sort of a filament that's been bent into a helical kind of shape, but this is more of a high frequency sort of staircasing kind of spiral pattern.
What the slide shows is basically yeah, it's basically that the scaffold itself starts spiraling, but the loops are still kind of random in their orientations, but they do remember that they're emanating from a scaffold that is spiraling. So if you look here, so this loops is shown in green and blue. They're emanating from the central staircase. We call this Leonardo staircase, like the staircase in Chambord. And but they do remember that they come from the spiral, though they are kind of randomish in terms of their orientation. What I didn't tell you is that there are two layers of loops. There are kind of bigger loops and small loops inside. So there's a whole lot of biology there, like condensing 1, condensing 2, nested loops. It's a whole zoo of things. But yes, the spiral seems to be rather maybe regular. We don't know. but it needs to be quite regular to explain high-C data.
Remarkable.
Yeah. And we don't know what kind of process leads to this, to spiraling itself. We know it depends on condensing 2, on one of loop extruders.
Nice.
One loop extruder, so activity on one loop extruder is not enough. You need so there are 2 loop extruders, condensing 1, condensing 2. Condensing 1 by itself does not lead to spiraling. Condensing 2 by itself leads to spiraling. It does not need the other one. Maybe it forms the spiral itself. Maybe it forms a filament. No, at this point, we don't know.
So I have a question about this spiral. So if I would imagine the spiral, I would imagine that, like, if you go around one winding, then you would have a peak in contacts, like exactly when you make one wind. But then I would also expect like the contacts to kind of be symmetric, whether you go like further up the spiral or back again. But do you see this kind of symmetry as well in the high-C data?
Yeah. You see the yes, the symmetry in high-C data is seen as the second band. I'm afraid of changing slides, but you do see this in the form of the second band. Let me, if annotation works, I will use this. Somehow it doesn't. But basically, yes, you see this symmetry because there is a second band. If you see my hands in high-C, so whether you go right or whether you go up, that's the symmetry in terms of directionality. And in fact, in some mutants, we even see the third and the fourth bands. We see several bands. But we need to get rid of some other factors that interfere with the spiraling. So this is still unpublished, but so we can still see high order waves in this, or oscillations, strictly speaking, in this data, which is really striking, given that it comes from real cells. Yeah. Looks like the spiral is very, very real. It may be changing its chirality as it goes, because again, it's an average over population of cells. Maybe in one cell it's a right spiral, in another cell it's a left spiral. Maybe halfway it's right, and then it changes and becomes left. So that's very possible. So there might be all sorts of defects in the spiral that we don't see because they average over in a pretty good into a pretty good spiral. Okay, let me maybe, okay, that's an animation. I don't know whether this would play. Looks like it.
I think also people are going to.
Okay. Yeah. I will just give a summary slide and that's it.
Sure.
Okay. And that's the summary slide, that chromosomes need a motor.
I think we should take this opportunity to thank both of our speakers who are still here. And Leonid for the tutorial. So thank you both very much. And for all the panelists and everyone who joined for this KNAW meeting. So thank you. People can stay on, but
thank you.
Close to
thank you for inviting me.
That was quite fun. Thank you, Leonid.
Thank you
both.
Thank you, Agnesa. That was really wonderful.